sometimes it blows my mind, ponder how they can figure any of this stuff out, because none of this is stuff that you can see with the naked eye. All these molecules are so tiny you can't even see them under a microscope, but somehow they not only found out these molecules were there, they can get down to the nitty-gritty of all the accounting for how the various levels of molecules that they all produce. There's that FADH2 that was made, making its 2 ATPs. Alrighty. Any questions up to this point? All right, well, let me point out a couple of kind of interesting things about this. Well, I guess I already told you about the pH differences. You can actually test the pH of the matrix versus the outer compartment and see that there is a pH difference between the two, which is what you'd expect from what we've seen in this process. There are some poisons that are harmful because they mess up the mitochondrial electron transport system. And just intuitively that makes sense. If you do something to harm this process, your cells are going to have less ATP than they're supposed to have, and that's going to harm you. One of the poisons that messes up this process is carbon monoxide. Carbon monoxide molecules look like this. They are a carbon atom and an oxygen atom joined together like this. And in terms of molecular shapes, the carbon monoxide molecule looks close enough to an oxygen atom that it can plug into the active site of this third cytochrome complex. Where do you find carbon monoxide? Well, one of the major sources are car exhausts. When you're running your car, some carbon monoxide comes out of there. And that's incidentally why they say don't ever run your car inside an enclosed space like your garage if you're doing repair work or something. The carbon monoxide levels build up to high levels. And they also say don't ever barbecue inside. Charcoal makes a lot of carbon monoxide, and it'll build up inside your house and get you. Anyhow, the carbon monoxide plugs into this active site right here, and it gets stuck there. It blocks it so molecular oxygen can't, or oxygen atoms can't bind there. Which actually reminds me of a vocabulary term. What do you call a molecule that slows down an enzyme by taking up its active site? It's a competitive inhibitor, right? So carbon monoxide is a competitive inhibitor for this oxygen binding site on cytochrome complex number 3. Here's another poison that relates to this. When Europeans were first contacting some of the Indians of South America, the Indians had an interesting way of fishing. They would dig up the roots of certain plants and grind them up into a paste, and then spread that paste out on the water, and it would stun the fish. All these fish would rise to the surface, and they could just collect all the fishes very easily. I think this is down in the Amazon somewhere. And when eventually some of these European scientists took these roots back and analyzed what exactly in the roots was stunning the fish, and it was a molecule named rotenone. And rotenone, they eventually found out, does this. Rotenone blocks this step right here, where the NADHs deliver their high energy electrons to cytochrome pump number 1. And so by blocking that step, well, it seriously reduces the ability of the mitochondria to do aerobic respiration. So the fish just got stunned from lack of energy in their cells. Actually, I just thought of a good exam question. I can't put it on now because I already wrote the exam, but if on the exam, if I ask you this, if you give a fish rotenone, or if you put rotenone in the water, how many ATPs per glucose would the fish be able to make? Give that some thought, see if you can come up with the answer. Remember, rotenone blocking NADHs from delivering any electrons. I think you're on the right track, but don't forget that these ATPs from the electron transport system are not the only ATPs that are made from the glucose. Don't forget these 4 ATPs as well. Well, let's think about it. So if we stop the NADHs from ever delivering high energy electrons to the electron transport system, we're not going to get that, and we're not going to get that. These 2 NADHs actually get turned into FADH2, so those can still function even in your rotenone poisoned fish. So we have 4 ATPs made there. And 4 ATPs made from these FADH2s make a total of 8 ATPs there from the electron transport system. Plus these 4 ATPs made in glycolysis and Krebs cycle. By my calculation, it seems like you're going to get 12, the fish are going to get 12 ATPs instead of their normal 36 per glucose. So they'll have an energy shortage enough to stun them, and that's how they collect them from the water. You buy it? All right. And one more little fun fact about the electron transport system here. The major function of this mitochondrial electron transport system is, of course, to make ATPs. You make the hydrogen ion concentration gradient, and they zoom back through the ATP synthase enzyme like that and make ATPs. In certain types of mitochondria, not in a lot, but in certain types of mitochondria, the ATP synthase enzyme has been modified so that it doesn't make ATP. Those hydrogen ions just zip right on through, and nothing is made. No ATP is made. And so you may think, well, what's the point in that? Why build up a hydrogen ion concentration gradient and let the hydrogens go back in and not get anything out of it? Well, if there's no ATP being made, the full kinetic energy of those hydrogen ions comes barreling right on through. And so you get a lot of friction as those ATPs, as those hydrogen ions crash into the other molecules in the matrix. And so it makes a heat generating type of mitochondria instead of an ATP generating mitochondria. And that is the body tissue that does that is something called brown fat, which this is a photograph of. You can see some mitochondria there with their highly folded inner membrane in that region of the picture. Anyway, babies have a lot of this brown fat on their body, and so it's a tissue that uses the energy, actually fat more than glucose, but it uses the energy to build up a hydrogen ion concentration gradient, but not for making ATP, just for generating heat. And then as we grow from babyhood to adults, we don't need as much heat generating tissue, and so we tend to lose brown fat as we get older. Goody? All right, so there's the big picture of aerobic respiration with the 36 ATPs per glucose. Now, the 3 steps that take place in the mitochondria, transition step, Krebs cycle, and electron transport system, all of those only operate if there's oxygen present. In other words, if there's not enough oxygen, all 3 of these steps shut down. But glycolysis, the step that's out there in the cytoplasm, does not require oxygen at all. It goes just fine, even if there's no oxygen around. Oh, actually, I got a little ahead of myself. come back to the oxygen in a second, but one thing I want to point out here is this. Here's a simplified diagram of the first three steps. Glycolysis is glucose going to pyruvates, and then in the transition step, the pyruvates become acetyl-CoAs, and then in Krebs cycle, the acetyl part of the acetyl-CoA gets fed into the Krebs cycle, and well, these are all parts of aerobic respiration. I mentioned at one point that glucose is the favorite fuel of these of cells, but if there's not enough glucose around, they can switch to other molecules as a fuel source. Fatty acids are their favorite alternative fuel. Matter of fact, a lot of cells in your body regularly use fatty acids from your blood. They can use glucose also, but they can take fatty acids just fine. And cells can also use amino acids, but they don't really like to do that unless they're really desperate for energy and there's no glucose or fatty acids around. What I want to talk about briefly here is exactly where those energy from molecules from fats and amino acids feed into the aerobic respiration process. Let's see here. All right, so remember, fats are a type of molecule called a triglyceride. And a triglyceride looks something like this. Each triglyceride is a glycerol molecule that's joined to 3 fatty acids, which maybe I'll draw like this. I'm leaving out a few atoms here because, of course, there are hydrogens, for instance, attached to a lot of these carbons. Well, if a cell is using fats for fuel, it detaches the glycerol from the 3 fatty acids, and the glycerol can be changed into pGal by an enzyme. And so the glycerol essentially feeds into the glycolysis pathway right there as pGal. The fatty acids themselves are then broken up into acetyl groups. There's an enzyme that chops apart the fatty acids into acetyl groups. There's an acetyl group right there. Here's an acetyl group right there. There's an acetyl group. There's an acetyl group. Acetyl group. Acetyl group. Acetyl group. Each fatty acid can be broken up into several acetyl groups, and then those link to coenzyme A, and then with the acetyl-CoA that feeds right into the Krebs cycle. With amino acids, there's 20 different amino acids. There's their general structure. And since there's 20 different amino acids, the biochemistry is a little complex, but many of the amino acids can be modified by enzymes to go into Krebs cycle intermediates. Like, if you remember the Krebs cycle, there was something called succinate and another intermediate called malate. The amino acids are turned into those molecules and then fed into the Krebs cycle. Okay, so I was about to say a minute ago, the glycolysis step is the only step of the whole process that does not require oxygen to occur. So if there's no oxygen around, transition step, Krebs cycle, and electron transport system just don't work. They just shut down. And as an example of when there's not enough oxygen for the mitochondria to function properly, think of bursts of activity. If you're sitting there relaxing and then someone says, hey, do this burst of activity, you have to lift the couch or something, or jump in the air, or sprint a short distance. During those short bursts of activity, your blood is not able to supply enough oxygen for aerobic respiration, and so your cells have an alternative method of respiration, which is the anaerobic respiration, which is sometimes called fermentation. I guess we're out of time. Okay, well, we'll pick up anaerobic respiration on Wednesday, and I will see you all in the lab today.